These are the lecture notes for chapter 29 on heredity, our last chapter. Okay, so starting here, it says genes, which are defined as segments of DNA, contain the recipe or blueprint for the synthesis of proteins. The expression of genes can be controlled by other genes, so whether or not they're turned on, whether they're turned off, that's what we mean by gene expression. So the definition for gene is a segment of DNA that codes for a protein. There we go. So it is a gene is a segment of DNA that codes for a protein. So that codes for the synthesis of a protein. That's what a gene does. That's the purpose of DNA, is to make proteins. Because our body is composed of so many proteins, that's the reason that we can also say that genes code for our traits. Genetics is defined as the study of the mechanism of heredity. A lot of this came from Gregor Mendel, although his studies he used garden peas and studied garden pea genetics and was able to apply a lot of what he learned to human traits. But like it says here, it says human traits are much more complex than that. There are a lot more, some human traits are inherited in the same way that the garden pea traits are inherited that he studied. But then there are some that are more complicated. Okay, so section 29.1, the vocabulary of genetics. If you have not had biology, you're going to need to really pay attention. If you have had general biology, then this should be somewhat of a review. But this is going to focus, if you took general biology and learned genetics, you did some of your topics were on like the genetics of garden peas or other organisms, but this is just going to focus on humans, of course. So all cells in the human body except gametes, and gametes are defined as egg and sperm. So all the other cells in the human body have exactly 46 chromosomes, which is the diploid number of chromosomes. So all of our body cells, except the gametes, have a copy of 23 chromosomes from the mother of the person and then 23 from the father. So in other words, that number 46 is really 23 pairs of homologous chromosomes. One pair of those chromosomes are sex chromosomes. If you get 2 X sex chromosomes, then genetically you are a female. If you get an X and a Y sex chromosome, genetically you would be a male. So one of those 23 pairs is the sex chromosomes, and you, just like all the pairs, you get one of them from your mom and one from your dad. The other 22 pairs of chromosomes are called autosomes. And again, you get 22 of those from your mom and 22 from your dad. So sometimes we look at all of those chromosomes. We take a picture of all the chromosomes. It's called a karyotype. So this is just a picture of all 46 chromosomes lined up in homologous pairs. So all 23 pairs kind of lined up. You can look at those and see if there are any abnormalities, any extra chromosomes, any missing chromosomes, that kind of thing. And then our genome is our entire genetic makeup. It's all of our DNA. And of course, half of that comes from our mom and half from our dad. This is a picture of a karyotype. Basically, what happens is to prepare a karyotype, part of it is it's all done with technology, but partly you have to have a microscope and you have to be able to visualize all the chromosomes in somebody's cell. So normally you can do that in prophase and even metaphase when a cell is in the process of dividing. The only time you see chromosomes is when that cell is going through mitosis, when it's in the process of dividing, because the chromosomes are all spread out and they're in a form called chromatin when the cell is not dividing. But basically you take a picture of the cell when you're looking under the microscope and you can see all 46 chromosomes clearly. You take a photograph. Then that photograph goes into a computer program, and you're able to manipulate and pair up all of the chromosomes. Pair 1, pair 2, pair 3, pair 4, pair 5. You're able to pair up all the homologous chromosomes, and then this is the 22nd pair down here at the bottom. And the 23rd pair, these are two X's. The Y is just half of an X, so that's how I know, because they're not duplicated, so this doesn't look like an X, but once it duplicates, it would look like the letter X, because there would be two strands connected at the middle. These chromosomes are not duplicated, so they don't look like the letter X, but this would be an X, and this would be an X, so this would be genetically a female. Now also in, let's see, this is pair 23, 22, 21. This is pair 21. If there were 3 of these, then this would be an individual with Down syndrome. So you can look for abnormalities and see if someone has a genetic disorder. All right. The different versions of genes are called alleles. So alleles are genes that occur at the same location on homologous chromosomes, but they can be different versions. I don't know, what's a human trait that I can talk about for an example? Let's say your mother gave you an allele for, let's see. There's a human trait called hitchhiker's thumb. Either your thumb bends back like a hitchhiker or it's straight. It's either hitchhiker thumb or straight thumb. So let's say that your mother gave you the allele for hitchhiker thumb and your father gave you the allele for straight thumb. So the gene is for the thumb trait. So both of those genes would be located at the same place on these homologous chromosomes, but they can be different. The alleles can be different. So alleles are just different versions of genes. So make sure that you know that. Alleles are the different versions of a gene for a trait. The DNA sequence can be the same or different on those alleles. So if we go back and talk about that, say your mom gave you hitchhiker's thumb and your dad gave you straight thumb, well, you could have had a different result. You could have had your dad gave you hitchhiker's thumb and your mom gave you straight thumb, or they both could have given you the same allele. They both could have given you the allele for hitchhiker's thumb, or they both could have given you the allele for straight thumb. So if they both give you the same allele, we say that's homozygous, that you're homozygous. That's when the alleles that you have from your parents are the same for a trait. But if you are heterozygous, that would be like if one parent gave you the hitchhiker's thumb and the other one gave you the straight thumb allele. Heterozygous means the alleles that you got from your parents are different for that same trait. Okay, so homozygous, the alleles for a trait are the same from both parents. Heterozygous, they're different. Now, these terms you need to know as well. If a gene or an allele is considered dominant, then if you are heterozygous, that's the one that's going to be expressed. So the dominant allele in our example is, it's funny because I didn't even know this trait that they were using for their example was a thumb trait, but it's not the same one I'm using. But let's say that you are heterozygous for the thumb trait that I told you about. One of your alleles from one parent is hitchhiker thumb and the other one is straight thumb. What you're going to actually have is both of your thumbs are going to bend back in the shape of a hitchhiker's thumb, because that is the one that's dominant. That's the allele that's dominant. So the dominant allele masks the expression of the recessive allele in heterozygous individuals. Okay? The dominant allele masks or suppresses the expression. I don't know why it's not highlighting. All right. The dominant allele masks the expression of the recessive allele in a heterozygous individual. So the example they gave here is loose thumb ligaments is a dominant trait, designated as capital J. Tight thumb ligaments is a recessive trait, designated as lowercase j. So the dominant allele is the loose thumb ligaments, the big J. If you have tight thumb ligaments, then you must have 2 copies of the little j, because that's the only way you would express a recessive trait, is if you get both alleles from both parents that are recessive. All right, so the dominant trait is expressed even if the other allele codes for the recessive trait. So if you have 2 big J's, or if you have big J, little j, you will have double jointed thumbs, in other words, that have loose ligaments. The only way you're going to have tight thumb ligaments is if you are little j, if your alleles are both little j alleles. Now, the other terms you need to know are, to really understand genetics and genetics problems, are genotype and phenotype. The genotype is going to be whether or not you're homozygous or heterozygous for the trait. So you would say like homozygous dominant, homozygous recessive, heterozygous. Those would be words you would use for genotype. It's the actual genetic makeup of a person. So if we use that double jointed thumb example, these are the 3 possible genotypes. 2 big J's or homozygous dominant, big J, little j, which is heterozygous, or 2 little j's, which is homozygous recessive. So these are the 3 possible genotypes when we're just looking at the thumb, the double jointed thumb example. The phenotype is actually whether or not your thumbs are double jointed or whether or not they're tight and they're not double jointed. So there are 2 possible phenotypes. If you have genotype, big J, or little j, they made a mistake. Let me fix it. Can you guys see the mistake? Can you see the mistake here? All right, so what we want to do is go up here to the little j, and change that. It should be big J, little j. So if you are big J, or big J, little j, you'll have double jointed thumbs because the big J is dominant. But then if you're little j, you will not have double jointed thumbs. Okay, so that's the difference between genotype and phenotype. Now we're on section 2, 29.2, sexual sources of genetic variation. There are 3 different events that can cause genetic variety in offspring. There's independent assortment, what we call crossing over, and random fertilization. Those are the 3 events that can lead to, besides just DNA mutation. But we're just going to talk right now about what these are: independent assortment, crossing over, and random fertilization. So one of the laws that Gregor Mendel discovered is the law of segregation. And you probably already know this information just because we tend to kind of grow up knowing it and learning it early on in life now. It's just something that everybody really kind of seems to have a good grasp of. And that is that for every trait, there are 2 alleles for each trait. One comes from the mom and one from the dad. And when the person's sex cells are forming sperm or eggs, when you're forming gametes, then those alleles will separate. Okay? So let's say that you have big J and little j. your gametes will either have big J or they will have little j. They're going to have one of the two alleles, and so those alleles separate in the process of gamete formation. Independent assortment happens when we're talking about two different traits at the same time, or more than one trait at the same time. So if we have on one chromosome a big B and a little b, and we have on another chromosome a big J and a little j, then each gamete is going to get one of the B's and one of the J's. Okay? One of the B's and one of the J's. So the gametes, your egg and your sperm, can either have big B, big J, big B, little b, big J, or little b, little j. Okay, this is the possible alleles that you could have in your egg or your sperm. Okay, now where independent assortment comes in is that it doesn't matter if you inherit big B, you still can inherit big J or little j. It doesn't matter if you inherit little b, you still can inherit big J or little j. So you get all the possible combinations. So it's whether you inherit big B or little b is independent of whether you inherit big J or little j. And you can actually calculate the combinations that we can get from independent assortment. So N is the number of homologous pairs, which is 23. So we can calculate it. In gametes, which are egg or sperm, we have 2 to the 23rd power, or 8.5 million possible combinations just due to independent assortment. Just due to independent assortment of traits, we have 8.5 million different combinations, which means that we have 8.5 million different varieties that are contributed to the offspring, that are possible in the offspring. Now, we're on slide number 14, so we'll start with slide 15 in the next recording.